we have to draw and label a diagram to show the fluid mosaic model of the biological membrane in this particular question. We've got a nice diagram question which people usually like doing, well I like doing them, so we can quickly draw a few points. So remember that the fluid mosaic model is a phospholipid bilayer. So that's the first part that you have to draw. We have to draw the polar heads here, as well as the non-polar tails. Once again, the non-polar tails, which are connecting to each other, as well as the polar heads. Okay? So, after that, then we've got an integral proteins as well. So these are proteins that are kind of stuck within the phospholipid bilayer, almost as if it was part of it. Okay? So this is our integral protein. It's called an integral protein because it's integrated within it, it's part of it. Sitting on top of it might be another peripheral protein. And sometimes within the phospholipid bilayer, you've also got like another molecule of cholesterol. I don't really know how to draw cholesterol, but if you just label it as cholesterol, then it should suffice. Okay? We've drawn a few things there. What else can we draw? So we can also draw a little glycoprotein on this peripheral protein. So this is a glycoprotein. A little glycogen tail. So it's a carbohydrate which sits on the protein. And another thing that we did forget to do is we had to talk about, to label these as the heads. So the heads are polar heads. And then the next one is these one here are the non-polar or hydrophobic tails. We can also label the thickness of it. That will give us an extra mark as well. So it's about 10 nanometers thick. So it's very thin actually. So this is a medium question. So it usually takes around about 6, maybe 7, probably usually around about 6 marks to get the full degree of to get 100%. So let's count them. So we've talked about the thickness, 1, 2, the integral protein, the glycoprotein, 3, 4, 5, and 6. So we talked about the non-polar and the polar heads. So that's usually enough to get you full marks for a question like this. So continuing on with another medium question, question number 2, we have to compare simple diffusion with facilitated diffusion as mechanisms to transport solutes across membranes. So every time that there's this action word called compare, then you want to do a table. That's the easiest way to do it. So let's draw on the left-hand side. We can talk about simple diffusion. And on the right-hand side, we can have facilitated diffusion. Okay, so let's talk about simple diffusion and facilitated diffusion and how they're similar. First of all, no energy is required. Okay, because you're going down from a concentration gradient from an area of high concentration to an area of low concentration. So we can write that down as well. Requires concentration gradient. So concentration gradient. And similarly for facilitated diffusion. Okay? What else is there? So we've talked about the similarities. Now let's talk about the differences. So simple diffusion goes directly through the phospholipid bilayer. whereas facilitated diffusion doesn't. It does not go through, does not directly go through the phospholipid PL bilayer. Okay? So because it goes directly through the phospholipid bilayer, it actually makes it slower. Think about it as a membrane, as almost like a bunch of thick spider webs. So you've got to kind of wade through the spider webs. That's relatively slow. Whereas with facilitated diffusion, you have a tube which you can go through. So it's like a tunnel. You can pop through the tunnel. You don't have to break apart those spider webs, and you can pop through to the other side. So it's much quicker. So let's talk about the speed. So slow speed, and this is fast speed. And I just mentioned before how we had to pop through a tube with facilitated diffusion. Facilitated means that it's being helped, and in order for these molecules to be helped through, they require a channel protein, which is completely different from the simple diffusion. The simple diffusion, no channel protein. Whereas for facilitated diffusion, channel protein required. So now we've got 1, 2, 3, 4, 5 different points. How about a couple, one or two more? So let's think about, how about the specificity? So for simple diffusion, anything can kind of go through, as long as it's small and it can pass through the phospholipid bilayer. So it's relatively non-specific. Whereas with facilitated diffusion, if you have a channel protein, and that channel protein is a certain size or a certain charge, only certain molecules can pass through. If it's a sodium channel, for example, only sodium will pass through it. Potassium or glucose, that won't pass through it. So it is very specific. The last thing to keep a hold of is to know that every single time that you do one of these compare questions, always give examples. So what's an example of some molecules that can progress through simple diffusion? So one example is oxygen, as well as carbon dioxide. Whereas over here, your example are usually larger molecules, so amino acids or sugars. And there you have it. There are plenty more YouTube videos for you to check out. Just click on the links below. If you'd like to download the questions, as well as the answers, make sure to like us on Facebook first. And finally, if you'd like to find out how I got a 7 in high-level IB biology, make sure to check out our website in the bottom right-hand corner. Thanks.